Thank you. So, thank organizers for putting me to the very end of this schedule. So I hope everyone is still awake. And I hope this is not the only presentation you will remember after the conference. Okay. So I know exactly what you want at this moment. So I'm going to keep my talk as concise as possible. So I'm going to talk about our effort at Schrodinger to develop novel methods to address challenging perturbations that are outside the domain of applicability of conventional methods. So as we all know, the original theory about FEP has been there for more than half a century. And since the initial application of FEP to calculate protein inclined infinity, people have been focusing on very small R group modifications and single atom mutations. For example, back in 1985, the Jorgensen group first applied FEP to calculate the solution finish difference between acid and methanol. In the following year, the McCommon group first applied FEP to calculate relative binding finish between benzamidine and fluorine benzamidine. And then in 1987, the Coleman group first applied FEP to study the effect of nitrogen to oxy mutation on their relative binding finish. And even now, a large portion of research is still focused on very modest R group modifications and single atom mutations. So I have these two examples here. So one from the Jorgensen group in 2014, the other from Macrocycle group in 2017. So both focusing on relatively small R group modifications and single atom mutations. And those are very important and common mutations we can encounter in drug discovery projects. And we have to acknowledge that it is definitely not easy to get those correct reliably. And at Schrodinger, we spend a lot of effort, both on the small molecule force field side and on the sampling side. And in 2015, we published this paper demonstrating that we can accurately calculate the binding finish changes for those modifications. And I think this plot has been showing in several presentations. So Woody just gave a comment about this plot. Is he still here? Yeah. So I just want to make a few additional comments to make it clear. So what we want to present in this plot. So first of all, I agree the R square value of all the points from all different targets is misleading because that depends on the experimental information. However, we only reported R squared and RMSE for each individual target, which are statistically meaningful. That's first point. Second is, if we want to do the selectivity, all you want is the binding affinity on different targets, which is similar to what we plot here. I'm not saying it's the same. Okay. Third point is about the null hypothesis. I think Guanglei gave a very good presentation yesterday about this topic. And if you want to discuss more about this topic, I think there is a very good paper here you can read more to see why the gas zero hypothesis is not a valid model. But this is not a focus of my talk. Okay. So after that, FE plus has been used prospectively in many projects, and a similar level of accuracy is also obtained in those prospective studies. I think this is a real testing about any method. And last year, we spent a lot of effort and collected the FE plus performance on, I think, about 100 projects we were able to collect data on. Those involves more than 100 targets with more than 3,000 ligands. And the overall MSE is about 1.1 kcal/mol. And the MUE for the vast majority of projects is less than 1, so in perfect agreement with the accuracy we're saying in the retrospective validation. And accurate calculation of finish change for R group modification and for single atom mutation already allows a large number of molecules to be tested. And we saw a great value already applying those methods. However, the impact of FEP is going to be limited if you restrict yourself to those modest, small modifications. Because in drug discovery projects, the chemists always want to explore a broader chemical space with more challenging calculations. For example, if the core of the molecule have certain enemy toxicity issue, then they want to do a scaffold hopping, for example, to change the size of ring, to introduce additional rings in the core of molecule. So how do you calculate binding affinity for core hopping modifications? Another example. So for drug targets that do not have deep buried binding pocket, blocking the protein-protein interface proves to be a very useful strategy. And microcycles are very good candidates to block protein-protein interface. So how to calculate the affinity to cyclize the needed molecule into a big macrocycle? Another example. So while a majority of small molecules bind to the target through unbounded van der Waals or electrostatic interactions, there is another class of small molecules that form covalent bond with protein residues in the binding pocket. So how to score covalent inhibitors? And lastly, so sometimes to improve the solubility or binding potency, or to balance other enemy toxicity issues, the chemists would like to consider perturbations that may change the net charge of molecule. So how to deal with charge perturbations? So over the past few years, we have been working on novel methods to address those challenges, and the talk is going to focus on the methods to deal with those very challenging perturbations. Okay. So first, about core hopping. So I mentioned earlier, sometimes the chemists would like to consider perturbations that change the size of the ring or introduce additional rings, the so-called scaffold hopping or core hopping modification. And so for R group modifications, sometimes you can easily revise the synthetic route to make them without too much trouble. However, for core hopping modifications, because the topology of the pharmacophore is modified, so it really takes a long time to synthesize molecule with a new core. And because our pharmacophore is changed, the majority of core hopping attempts failed to deliver any valuable matter to the project because most often it's not potent enough. But even that, core hopping is often attempted in discovery projects because very often it is the only viable strategy to address a particular enemy toxicity issue. And also the lower IP generated by core hopping makes it even more attractive. So therefore, if we can do accurate French calculations for core hopping modifications, then we can enable the chemists to pursue core hopping attempts with confidence that the resulting molecule will be potent for the project going forward. Okay, a little bit theoretical consideration. So compared to conventional R group modification FEP, well, you can see the bounded stretch interaction between the leftover dummy atom and the physical atom won't affect the ensemble of configurations for the physical molecules. In core hopping FEP, this is different. So for example, showing here is a mutation to cyclize linear molecule into a fused ring. If you retain all the bounded stretch interaction between the leftover dummy atom and the physical atom in the ligand, then you can see that the ensemble of configurations sampled for the mixed topology molecule is going to be different from the ensemble of configuration for the real physical molecule. And as we shown in this paper in collaboration with Professor David Mobley, if you don't deal with this correctly, there are large errors introduced in the physics results. So therefore, in core hopping FEP, you have to explicitly calculate the affinity to break a bond or to form a bond. And this was believed to be extremely difficult because in classical mechanics force field, we usually use harmonic potential to represent bounded stretch interaction. But the harmonic potential approaches infinity when the distance of atoms is infinity, causing singularity and numerical instability problems in FEP. So to address this difficulty, we have designed this lower functional form called soft bound stretch potential with functional form shown here. So without too much mathematics, you can easily show that when lambda equals 1, this potential recovers harmonic potential. And when lambda equals 0, this is 0. And for any value between 0 and 1, the first and the second derivative of the potential with respect to lambda and r are continuous. So we can do MD simulations using this potential. And in this paper, we prove that by using this functional form, we can rigorously calculate the finish to form or to break a bond, enabling accurate finish calculations for co-hopping modifications. So here's one example showing that we can calculate the finish for ring opening closing perturbation. The receptor is EZH2. So the 4 molecules shown here are close analogs of the development candidate in clinical trial. So here 22 and 27 have a fused ring, 29 and 31 have a single ring, and mutations from 22 to 29 or from 27 to 31 involve the ring opening perturbation. And the calculated binding affinity results agree well with experiment. And one very interesting feature about this SCR is the strong non-additivity effect. So I think many speakers talk about non-additivity in their presentations. So in particular, in the presence of the 5-member ring in the terminal molecule, opening the ring only modestly increased binding potency by about 0.6 kPa. In comparison, in the presence of 6-member ring, opening the fused ring significantly increased binding potency by about 1.9 kPa. And this strong non-additivity effect is accurately calculated by co-hopping FEP. And we can also evaluate very complex ring structures, like the speaker just mentioned, have a bridged ring. So we can evaluate that accurately. So here's one example. So for base 1 inhibitors, introducing additional ring to form this very complex bridged ring, the affinity was accurately predicted. We can also evaluate ring size change perturbations, for example, going from a 5-member ring to 6-member ring. In this case, for E alpha, expanding the ring decreased binding potency significantly, and we get that correct. And moving the carbonyl group around the 6-member ring from here to here further decreased binding potency. Experimentally, this is an inactive compound, and all this is accurately predicted by FEP. And we have validated this method on 6 different targets with many different unique co-hopping modifications covering a diverse chemical space. Again, the accuracy is perfect. RMSE is about 0.5 kPa. So I'm going to show one example where the large-scale application of co-hopping FEP led to the discovery of a novel chemotype in a very short period of time. In this project, the collaborator became very interested in a development candidate that was recently sent into the clinic. And that compound have a sub-nanomolar in vitro potency. And all of the obvious, like R group modifications, have been covered by existing IP. I think this is very common in this field. So then we use computational tools, enumerated 100 synthetic feasible novel cores, and then use FEP, co-hopping FEP, to calculate the binding affinity for those 100 molecules. And based on the calculation, 6 of the molecules were flagged to be sensitized, and then the collaborator managed to sensitize 3 of the compounds. And here's the results. So it turned out all those 3 compounds are indeed very potent, and they were not covered by existing IP. And further modification of molecule Z led to the discovery of a development candidate that is currently under consideration. So I have note here that using co-hopping FEP, the whole process only took 3 months. By comparison, the brute force chemistry effort will take much, much longer time to achieve the same goal. I think this is a very good example demonstrating the impact or the value of accurate finish calculations to speed up drug discovery process. Then I'm going to talk about microcycles. So we know that microcycles occupy a unique region in chemical space, bridging between small molecules and biological molecules like peptides, proteins. And in general, they are more difficult to make. However, due to their unique ability to balance various properties, including potency, selectivity, membrane permeability, and bioavailability. Microcycles are becoming a very important drug class, particularly for targets that are difficult to drug by small molecule, like protein-protein interface I mentioned earlier. So in microcycle drug discovery, the most important question is, why is there a microcyclization strategy to cyclize the linear molecule into cycle will improve the binding potency? And what is the optimal linker size and chemical matter for cyclization? And both those two types of perturbations are very difficult for conventional FEP method. So by using the same soft core stretch potential I mentioned earlier, and adjusting the protocol specific for microcycles, we have extended FE Plus to support microcycle perturbations. So one example. So here the receptor is MTH1. In this case, cyclizing the linear molecule into microcycle increased the binding potency significantly by about 4 and 5 kcal. And this is accurately calculated by the FEP method. Another two examples. First case, for a pair of base 1 inhibitors, cyclizing the linear molecule into microcycle increased binding potency by about 2 kcal. In the second case, for a pair of CK2 inhibitors, cyclizing the linear molecule into microcycle actually decreased binding potency by about 2.7 kcal. And both effects were accurately predicted by FEP. And very importantly, in addition to numerical agreement with experiment for the predicted binding affinity change, from the simulations we can also give physical insight as to when and why microcyclization can improve the binding potency. So here's the MTH1 example I mentioned earlier. Here, cyclizing the linear molecule into the cycle increased binding potency by about 4 kcal. And we can explain where this 4 kcal comes from. So showing here the red and the blue curves are the distribution of this key torsion sampled in solvent and complex. So the upper panel is for the linear molecule and the lower panel is for the microcycle. So by comparing those two plots, you can easily see that the two molecules sample roughly the same conformational space in the complex. However, their distributions in solvent are dramatically different. In particular, while the linear molecule can sample many different conformations in solvent, cyclization limits the amount of conformation space in solvent, reducing the amount of entropy loss experienced upon protein binding, leading to the improvement in the potency. And we can also evaluate ring size change perturbations like small molecules. Here's one example. So for 4 CHEK1 inhibitors, so with different ring sizes from like 4 carbon linker to 5 carbon linker to 6 carbon linker, the calculation agree well with experiment. We can also, many other examples, I don't talk in detail, and we benchmark this on 7 different targets with many different unique perturbations. Again, the RMSE is about 1 kcal. And I think yesterday, Katerina from Bayer showed one example in their results. So they have been using a pre-released version of our method and applied on a few microcycle systems, also obtained very good results and published this result in this paper. Okay, about covalent inhibitors. So covalent inhibitors constitute a significant fraction of approved drugs in the market. And due to their unique ability to achieve very high potency, selectivity, and good pharmacodynamic properties, covalent inhibitors is emerging as a very important drug class for certain disease areas. And the development of covalent ligand FEP was motivated by a research collaboration with Roche. So in this project, the goal is to improve the binding potency of an initial lead compound binding to HCl receptor. So the lead compound have a cyano group that forms a reversible covalent bond with the cysteine residue in the binding pocket. And the task is to identify building blocks from a library about 3,000 fragments on this R position with improved binding potency compared to the initial lead compound. And FEP plus scoring was compared head-to-head with the 3 other methods available to the chemists, including the selection based on their experienced medicinal chemists, or from the internal docking, or from a docking plus manual inspection by an academic lab with expert knowledge on these systems. And all the 4 groups are allowed to submit 10 recommendations and 5 backups before a predetermined deadline. And then the molecules are made and tested after that, so it's truly prospective. So the thermodynamic cycle of reversible covalent inhibitors shown here. So first step, it forms a non-covalent complex, and then the chemical reaction happens. So if the reaction is reversible, then the binding affinity is determined by the relative affinity among those states. So if the equilibrium concentration of the non-covalent complex is much, much lower than the covalent complex, then we can ignore the middle state. So then by calculating the affinity to mutate from protein ligand 1 complex into protein ligand 2 complex, and the affinity to mutate from ligand 1 to ligand 2 in solvent, we can calculate relative binding affinity between the 2 ligands as the formula shown here. So the 2 legs in the FEP for covalent binding is shown here. So in the complex leg, protein ligand 1, covalent bonded protein is mutated to protein ligand 2, and then solvent is similar. So we applied glide docking and MMGBSA and identified 92 molecules for further analysis by Fe+. And based on the Fe+ calculations, we recommended 10 molecules plus 5 backups. And then the collaborator made 10 of the compounds selected by FEP, and also 10 compounds from each of the 3 other methods. Okay, this is important. So here's the results. So 8 out of 10 recommendations by FEP indeed bind stronger than the reference. By comparison, all the 3 other methods available to the chemists, they only identified 1 potent binder compared to the initial lead compound. And the common compound identified by 3 methods, compound 3, is an obvious potent binder based on the existing experimental SCR. And also, the structures selected by FEP is very diverse. I think this clearly speaks to the power of accurate prediction in affinity to speed up lead optimization process, particularly to improve the binding potency. Okay, so in the last few minutes, I'm going to briefly talk about our effort to do charge perturbations. So as mentioned earlier, so sometimes in the discovery project, to improve the solubility or to improve the potency or to balance other enemy toxicity problems, the chemists we may consider mutations that change the net charge of the molecule. For example, for a pair of PDB1B ligands, replacing the charged ring by a neutral moiety increased binding potency by about 1.8 kcal. So we all know that the periodic boundary condition in MD simulation introduces artifacts in the long-range electrostatic interaction calculations. And I think back in 2014, many speakers have talked about their method to deal with charge perturbations, including Benoit Roux, Gabe Rocklin, and a few other folks. So over the past few years, we have been working on systematic approach to address those charge perturbations and developed our unique protocol. So our method is similar to what many people have tried before. The idea is very simple. So for perturbation between a neutral molecule and a charged molecule, we introduce a very special molecule in the system. So the charge of the special molecule is 0 for the neutral ligand state, and the charge for the special molecule is plus or minus 1 for the charged ligand state. In other words, when mutating a neutral molecule into a charged molecule, we simultaneously turn on the interactions between an ion of opposite charge with the environment. In this way, the net charge of the two physical endpoints is 0, and the artifact is largely removed. And in practice, this is achieved by changing a water molecule into a sodium or chloride ion. So the purpose of using water is to avoid this special molecule in the neutral state getting stuck in hydrophobic environments. This is particularly important for membrane-bound proteins because the hydrophobic particles can easily get stuck in lipids. So for the PTB1B example I showed earlier, so we applied 4 different methods on this system. So the first method is the regular FEP without any special treatment on the electrostatic interaction energy calculations. Second is the charge that uses the PB correction to correct the artifact in the long-range electrostatic interaction calculation, similar to what Steve Rocklin and David Mobley introduced in their paper. The third method uses the solvent electrostatic offset potential correction, similar to what Professor Benoit Roux introduced in his paper. And the last one is the air chemical water approach I just mentioned in the previous slide. And for each method, we tried 2 sets of experiment. In the first set of experiment, we only have counter ion to neutralize receptor. In the second set of experiment, in addition to the counter ion to neutralize receptor, we also put additional salts to mimic the ionic strength of the buffer solution. And the dashed black line is the experimental value for this population. So we can see that if you don't have any special treatment, because of the artifact in the electrostatic energy calculation, you have very large box size dependence, I think which many people have already know this, not very surprising. And also, the additional salts in buffer solution have a big effect on the calculated binding affinity. And this is well captured, studied experimentally. By comparison, using all the other 3 methods, so the box size dependency problem is largely removed. And also, the results from 3 methods are within the statistical laws of each other. And the results with the additional salts agree well with experiment. Those are the solid line. And then we tested the same protocol on a large data set with more than 10 targets, with more than 30 unique charge populations. The summary is shown here. The overall RMSE is about 1.1 kcal/mol, comparable to the accuracy for the neutral populations. So in summary, so starting from 2015, after we making the R group modification and single atom mutation populations accurate and reliable, we have been doing more research to develop novel methods to push the boundaries of the finished calculation methods to enable accurate calculations for the following very challenging populations, including co-hopping modification, microcycle populations, covalent bonding, and charge perturbations. And both the retrospective validation and prospective applications of those methods have demonstrated their high level of accuracy and reliability. And then with this, I'd like to acknowledge the very long list of people who have contributed to this work. And thank you all for staying so late to listen to my talk. Thank you.
Questions for Lingle?
Yeah.
So on your core hopping scheme, where you're doing these softened bonded terms, are you also having to come up with softened angles and softened torsions as well, or is it just these harmonic bonds that are letting you do this?
The bond stretch are most important. The angles and the dihedral angles, softening those potentials also helps, but it's not that critical because you have a fixed boundary. The potential won't go to infinity.
Christopher? when you grow or annihilate the water for your charge thing, if the water's far away, you'd be growing or disappearing an enormous dipole. If it's close, you'd be growing or disappearing a smaller dipole. So was there any, I'm just wondering, would there be any effect of having this large or small dipole? Would it make a difference whether the water was close or far away?
Yeah, so the problem you just mentioned probably is relevant if you only have one special charged ion. But the trick is here, you have many more ions than just that special molecule. So the overall effect is smeared out.
150 millimolar salt
Yeah.
made such a difference?
Yes.
I see. Okay, thank you.
Yeah. This is really exciting work. I'm super excited about every aspect of it. But the loop closure for these macrocycles is really puzzling to me. So entropy is this
It's
mysterious
puzzling
thing.
to me as well.
And
Initially I didn't suspect it would work so well, but the results just came out so great. I don't know. I don't have an answer to.
Specifically, you had a mechanistic explanation involving these torsion angles, right? But if you look at those torsion distributions, there's no way that difference in population could explain a 4 kcal per mole difference. So what are we missing? What's the mechanistic explanation? I mean, you see a little bit of shifts around, but maybe it's a factor of 4.
So I don't know why you say this is a little shift, but not a big shift. Comparing the two red curves, not only the number of conformations are different, also the relative population is quite different. So you have to pay a large penalty.
Yeah,
to
that peak on the right? The peak on the left in the upper plot, that's not 4 kcal per mole. That's
we can quantify that. We can do experiment to quantify that. You can do very, very long sample and use, for example, the entropy, like for example, using the Shannon entropy formula, the rigorously calculate the entropy. We can do that. I think for this particular case, I'm convinced it's coming from the entropy.
interesting. Okay.
But the torsion is not all the entropy. That's just
Yes, and also
the 10 or so degrees of freedom collapse under 1 degree.
And also, you have to remember this here. I'm going to only showing one particular torsion, which is important. And there are so many torsions that contribute to the entropy, right? They add up together.
Well, there could be also internal strain on the acyclic molecule.
That's reflected in this population.
So this internal strain is not just a product of reducing entropy. That's an enthalpic contribution which is paid from the pre-organization forming the macrocycle.
Yeah. So here I'm showing the population. So whatever internal strain energy will be reflected in the final population. So that's not. I think it contributes, but it's already reflected here.
I think trying to do anything that just 1 degree of freedom is not going to give you any meaningful results. You have to learn it more.
Or you can do the final temperature difference to calculate the energy as a function of temperature. So you can.
Right, that includes the entropy, but it won't be able to explain why the entropy.
Yeah.
You can depopulate that population by putting a barrier there, right?
That's true. Yes.
And for this particular problem, is this looking at the case where it's the protein-protein binding and interfering with the protein-protein?
This case is not. It's just the same. Yeah.
Okay.
I have a question about the covalent bonding. In the thermodynamic circle you showed.
Yeah.
So how do we know that the intermediate step, if the concentration is really low, it can be neglected? How do we know the concentration was really low for the intermediate?
Yeah. So you can also run the FEP calculation for this lag and this lag, and then you can like get an idea about the relative affinity between those two states, and you can see the difference is really dramatic. It's more than 4 kcal. So basically means the population of this is
Yeah, but that
neglectful.
will probably depend on the warhead you're using, right? The warhead
Yeah.
you're inspecting.
So in general, I think you can do additional step to explore the relative affinity between those two states. But in this particular case, we validated this is not an important state.
Right,
Yeah.
but system dependent. So why still need a testing?
We have time for one more question because I want to ask a question and I'm moderator. So I'm really impressed by your the small size of the error for the absolute change in charge. I guess You mentioned a paper by Gabriel Rocklin and David
Yeah.
Mobley, and
Yeah.
I think what they showed and other people have seen is salt bridges tend to be over-predicted. So did you see any systematic? I mean,
Yeah.
Your results are so good. Does that mean we don't need polarizable force fields?
So yeah, we do have evidence for perturbations that break the salt bridge. It is actually more difficult, and the error bars are larger. But in this case, I think the majority of them are partially salt exposed.
Okay.
So even if you have a salt bridge, you have other interactions that compensate that effect. Yeah. But I do believe that polarizability will play some role for the some particular chemical groups. Yeah.
Great. Well, let's thank Lingle, and then